You can look at your agenda, and it will tell you which salon ballroom that you should go to, depending on the age of your child or the breakout session that you're in. So don't come back here. Okay, go past here and down to the left. All right, so our last morning talk will be executive function and cystinosis with Dr. Anna Francisco and Sophie Molholm. And they are at the Albert Einstein School of Medicine. That sounds really smart. I mean, I haven't heard of any Kevin Partington School of Medicine, so Albert Einstein's a much better name. And I would just say our daughter, and I know several other, I know Lars and Sam went to this study, and so did Jen. And I think actually Hadley did too, yeah? No, was going to. Okay. So anyway, be interested to see what they found over the last year. So welcome up. You on a solo mission? Oh.
Hi, good morning. Oh, can you hear me? No? Better? Hello? No.
Check.
I think it's, oh, there you go. Now good? Okay. Okay, so first, just thank you, Kathleen. I don't know where you are, but that was an incredible talk. Thank you so much for teaching us. And thank you for the introduction. I will be giving the talk, but Sophie is also around somewhere. Oh, there she is, waving. Okay, so today I'll be talking about executive functioning in cystinosis. And so this is work we've been doing with the generous support of the CRF. So just thanking the CRF for the support and also for having us here for the first time. It's so nice to see so many of you and meet new ones. Okay, so as I said, this is work I've been doing with Sophie Molholm at the Albert Einstein College of Medicine, which sounds super smart, I agree. But also work that I've been doing with John Fox and our Osnos, who are pictured there. Okay, so the work I'll be presenting today is actually part of a larger project that we've been working on for the last couple of years. There's a lot of stuff involved, so we have different groups that we are comparing, group of people with cystinosis, but also a group with people with chronic kidney disease and controls and the parents, and we are looking at different measures and using different techniques. But today I'll be focusing on preliminary findings on the comparison between a group of people with cystinosis and age-met controls, and using EEG, which I'll be talking a little more afterwards, looking at different components of executive functioning. Okay, so just starting by defining executive functioning, what are we talking about when we talk about executive functioning? So we can think about executive functioning as this set of control processes that really are responsible for these goal-directed behaviors that really help us optimizing our performance on usually complex cognitive tasks. So these set of processes, they allow us to be flexible and to adapt to new and ever-changing circumstances we encounter in our daily lives. So executive functioning has a lot to do with regulation at different levels. So we can think about cognitive and emotional and social regulation, but we can also think of this set of real life skills that executive functioning can really impact. So we can think about academic behaviors and work habits, and focused and engaged learning, planning and organizing, emotion and behavior regulation, conflict resolution and problem solving, and understanding social cues. So as you might have guessed from this very complex definition and set of components, executive function really is many things. So today we're going to be looking at 5 of those things, or 5 of those components. So I'm going to be talking about response inhibition, error monitoring, memory updating, set shifting, and conflict monitoring. So let's start. Okay, so as I probably said at the start, these are preliminary findings. So we're still completing some of our groups, definitely still completing some of our analyses. These are not our final numbers, and not all the interpretations are clear at this point, but we wanted to share with you what we're seeing as of right now. So the data that I'm going to be presenting are from a group of people with cystinosis, 34 people, a wide range of ages, as you can see, between 9 and 38 years old, and the group of people without cystinosis, kind of age matched, but we're still working on that. Okay, so before we go on to really look at the data, just wanted to explain you very briefly what we'll be looking at. Okay, so for most of our work, we use electrophysiology, EEG. Some of you here, I know a lot of you, a lot of the kids have done it, but also some of you have come to New York, so they'll know what I'm talking about. But basically, the participants come in and they are doing these computer tasks while we are measuring their brain activity, their electrical brain activity. So I'm not sure you can see really well, but we are rocking these really cool caps. They look like swimming caps, and they are full of these wires, and these wires are measuring our electrical brain activity. We also eat a lot of lollipops in the lab. Once the participant is doing the tasks and we are measuring their brain activity, this is kind of the type of wiggles we are actually seeing. So we don't see a lot while we are doing the data collection. But then once we process the data and it goes through a lot of cleaning and all of that, this is something that we can be able to get at. So I'm just showing you this so that we understand what type of plots I'll be showing later. So basically here, let me see. Does it work? Okay, maybe you can see my mouse, right? So we have time here. So basically how quickly our brain is responding to whatever we are showing. And here we have microvolts, so how big that brain response is to whatever we are showing. And you can see that we have all these kind of peaks, and they have these numbers and letters. It doesn't matter too much, but each of these peaks have been shown to kind of be associated with a specific cognitive process. So we kind of know what they are reflecting. Okay, so this is the type of thing we'll be looking at from now on. So starting with conflict monitoring. So in life, we are often faced with situations in which two or more competing responses or action alternatives exist. So detecting that conflict and then adjusting quickly will result in better performance. So to measure conflict monitoring, we use the task where we have these congruent or no conflict and incongruent or conflict trials, right? So basically the participant is seeing these arrows on the screen, and either it's a pretty easy task, because the idea is that you have to decide in which direction is this middle arrow pointing. So either we see it in the context of arrows that are all pointing in the same direction, so we are able to pretty quickly identify where they are pointing, or we are seeing them in this context in which they are pointing in the opposite direction of the arrows that are surrounding them, which makes it a little harder for us to make that decision because we have to deal with that conflict. So what we find, what we see in controls is what we should be seeing, and I highlighted the areas where you want to look at, so you don't have to look at all the wiggles that are around there. So if we look at this area here, and here we can see in the darker color, we have the no conflict trial, okay? So it was pretty easy. The arrows were all pointing the same direction. And in the lighter line, we have those trials that were a bit more difficult. So what we expect in this case is that the lighter line goes a little more down than the darker line because it's more effortful, right? So we're seeing that in control. So what we see in cystinosis. So looking at the same time window, we see this very similar pattern, right? And this is something that we've been seeing in our cognitive and brain function results, is that as a group, people with cystinosis don't differ too much from the controls. So their brain responses and their behavior is still very similar to that of controls. Sometimes it takes them a little longer to accomplish whatever the end results might be, but things are very similar. And you see it here too. So if we just take a look overall at the brain waves there, they're super similar, right? What we're seeing though is that it seems like this difference between the conflict and non-conflict is a bit larger for the cystinosis group. And that is mainly being explained by the response to the trials that have a conflict, so the harder trials to process. So it could be that people with cystinosis just have a little bit of a harder time to process that conflict, to work through that conflict that is being presented. Okay, so moving on to memory updating. So we can think of memory updating as the screening system that reviews information based on how important that information is, eliminates unnecessary information, and then replaces it with more relevant one. So it's something that is constantly happening kind of the background. So for this one, and this task is super difficult for those of you here who've done it, I'm sorry, I struggle too, particularly with the harder conditions. So this is a task with 3 different conditions. So we have a pretty easy one. Basically, every time the participant sees a square on the top left corner of the screen, they press a button. So that's pretty easy, right? A bit more difficult condition. Every time the square is in the same spot as it was in the trial before, you press the button. Now, the one that is impossible, at least for me, is you press a button when the square shows up where it showed up 2 trials before. So you have to keep keeping in memory where the squares are to be able to do the task, right? So what we see, again, in controls, very typical results of what we should be seeing, we see that there is this difference between conditions. So the easiest one really is no memory, right, involved, because you just need to press every time you see a square in that specific position, so you don't have to keep having in your mind where the square is. So that's pretty easy, so it's up here. And then the more difficult the task is, the lower the line goes, right? So what do we see in cystinosis? Again, pretty similar, right? We're seeing the same kind of pattern is there, this differentiation between the conditions. But interestingly, what we are seeing is that whereas for the easiest one where no memory is involved, it's really very similar between controls and cystinosis. We see that people with cystinosis are kind of processing similarly the harder conditions. So it seems like already for the one back condition, the one in which memory is already involved, but it's still not as hard as the 2 back condition. It's already being pretty difficult for people with cystinosis, or at least it seems to be more difficult than for people without cystinosis. Okay, set shifting. So set shifting represents the ability to jump from one task or mental state to another and back again at speed. So it really allows us to respond flexibly to whatever is thrown at us, and really is very important for learning and problem solving. So this task, also not an easy one, but basically you have these things will be presented in 2 colors. Here in this example, if things are presented in purple, so it will be numbers and letters. If numbers and letters are presented in purple, you are asked to think about the letters, to categorize the letters. If they are in red, you have to categorize the numbers. So for the letters, you have to decide whether they are a consonant or a vowel, and for the numbers, whether they are even or odd. And you have to, sometimes you'll have to switch, right? So you'll have to, in one trial, do the numbers, and then the next trial, do the letters. And other times it's a repeat. So you have a couple where you are doing just letters, and then you switch and you are doing a couple where you just have to think about the numbers. And this difference, because we really are interested in the set shifting, here we are really looking at this comparison between the moments where you are repeating your action and those where you have to switch your mindset to kind of do a kind of a different task. So here is what we see. And in light pink, we have those trials where we have to switch. So we had to make that little extra effort to think about a different thing or in a different way. And in darker pink, the repeat. So it was a bit of a easier process to do. And what we would expect really is this differentiation between the two. So the lighter pink here, so when we have to switch, it will be a more effortful process. So a higher wave there when compared to the darker, the repeat pink. So in the cystinosis group, again, you can appreciate how similar the brain responses are between the two groups. But there's here a time window, again highlighted, where things seem to be a little different. So we can see that there is this peak in the controls that is not there in the cystinosis group. There's still this differentiation between repeat and switch, right? So one is still being more easy to process than the other one, but there seems to be something different. Now, it's hard to say exactly what it means right now. For that, really, we need to look at behavior and also at all our standardized tasks that we are doing that really will help us understand how people with cystinosis are functioning in terms of behavior in set shifting. So still more to come on this, but anyways, there seems to be something that is a little different between the two groups later, so around 400 milliseconds, so half a second after the trial is presented. Okay, moving on to inhibition. So response inhibition really allows us, well, to inhibit our impulses and natural or dominant behavioral responses. And this, of course, in order to be able to select a more appropriate behavior that is more consistent with what we want to accomplish. And I'm immediately defining error monitoring too, because we use the same task for these two things. So really, error monitoring is about not only detecting an error that we just made, but also the ability to kind of adapt our behavior and adjust our attention so that we can avoid making further errors. And really, it relates to how we respond to our own performance. So for this, we used a task in which basically the participant is just looking at pictures on the screen and they have to press a button every time they see a picture. But if the picture is immediately repeated, they have to inhibit that response. So they shouldn't press the button. This seems easier than it actually is because, as some of you will know, we present this task very fast. So the images come up at you very fast. And there are many more trials in which you are supposed to press than the ones you shouldn't. So you kind of get used to be constantly clicking and then you have to stop that action. It's pretty hard to do. Okay. So when we look at inhibition, actually, there is no difference between the groups. So the performance is very, very similar. People with cystinosis seem to be as good inhibiting their responses as did the controls. However, when we look at error monitoring, there was a clear difference. So people with cystinosis were showing a smaller response, a smaller brain response to their errors. Now, we know from the behavioral response that we have that it's not like they were not aware of the errors. So they were still slowing down their response, for example, after they made an error, which really indicates that they were aware they made a mistake. What we think was happening here was that this really has to do with slower processing speed that we see very commonly in people with cystinosis. So they were processing it slower, but still processing that they made a mistake. Okay, so as a very, very short summary of the findings I just showed, so it seems like response inhibition is pretty much the same when compared to controls, but in all other components that we've tested so far and with the tasks that we use, there seem to be slight differences between people with cystinosis and people without. What the functional meaning of those differences are, we're still trying to figure out. And the results and the data that we haven't analyzed yet will definitely help us understand what those functional, the functional impact is. Okay, but just moving on to, as we know that there are some slight differences, I just put together some kind of suggestions to keep us on path in terms of executive functioning and how to help being able to plan and organize and accomplish the goals that we set ourselves to. So there are these kind of steps that we can follow. So we start with goal setting, of course. So the initial decision about the choice of a goal to accomplish. So what do I need, or what do I want to accomplish? And then a step of self-awareness. So recognize one's stronger and weaker abilities. So how easy is it going to be for me to accomplish the goal that I'm setting myself up for? The stage of organizing and planning. So what are the steps that I need to be completing in order to accomplish my goal? And how long will it actually take to complete the task at hand? And then flexibility and strategy use. So identify possible problems that may arise in the process. How can they be solved? And develop that flexibility in getting to alternative plans if the initial plans fail or they need to be changed. And finally, monitor and summarize. So assess progress, review the goal, review the plan, what was accomplished, did it work, did it not work, why was that the case, and can we be more effective in the future, and how? So getting into more concrete strategies we can use, and this is a bit more tailored to school environment, but we can very easily think about these in work environments too. So repeating instructions of new information so that more information can be captured. This is particularly important when we have working memory difficulties, right? Teaching these like self-initiated comprehension checking strategies. This is basically making lists and being able to check things from the list so that we can keep on track. Very important, providing a quiet, stable learning setting and reducing distractions in the environment, particularly, and a significant number of people who came visit us do have some difficulties with focused attention and keeping that attention for longer periods of time. Teaching and modeling self-regulation, practicing distractor blocking and attention switching, and providing extended time for tests and assignments, particularly to compensate for these processing speed differences that we are seeing very commonly in people with cystinosis. Just to end, so when I moved, I started being involved in cystinosis projects when I moved to the US kind of 7 years ago. So I've been very fortunate to kind of see the participants throughout the years. So a lot of people who came early on have returned. So we've been able to kind of start looking at these longitudinal differences in terms of cognitive and brain function, which we think can be really meaningful to the community. So we have here 2017 and 2022. So today I'm just going to mention some results on cognitive function. I didn't bring the brain results. And this is just ending. I think overall, what we are seeing are good news, but wanted to end with a very positive note. So this is a lot of stuff in here, but basically we have IQ here on top. So verbal and nonverbal, more spatial type of intelligence. And the bottom, we have measures from two different tests, a test called DCAFS that is basically executive functioning type of assessment, and the CPT, which focuses on attention and vigilance. And what you have to know so that you don't get confused by what I'm concluding about this is that whereas for the IQ, the higher the better, for these two tests, the lower the better, okay? So what we see, and this is here is a very mixed sample. So we have children, we have adults, so everyone is mixed here. We don't have, I think for these analyses, we have around 17, 18 people, so not as many as for the full sample, because of course no one has returned, or we have some new participants. But the good news and what we are seeing is that actually most people with cystinosis have kept stable or actually improved their performance during these 5-year time period. And that's it. Thank you for your attention.
Thank you, Dr. Francisco. Denise just asked me a question that I'll ask you. Are you still looking for participants?
We are finishing up our data collection, which is exciting. So we can talk later, but yeah, we're finishing up our collection of data.
Okay. What's the next steps?
Yeah, of course. So two parts to this answer. One I'm able to answer, the other one not so much, but I'll still tell what my thoughts are. The first one, still regarding this data, there's still a lot we can do. You know, we have more data to analyze. We didn't do only EEG, we've also done cognitive testing, as you know. So we have really a rich set of data that we can still tap into and start really relating our brain data to what we really see in terms of behavior and outcomes, which I think is really exciting. Something else that I'll be working on, hopefully, is on these guidelines for schools and parents that are derived from what we are seeing in terms of executive functioning. We were really interested in executive functioning, particularly because a lot of the conversations with the families and with people with cystinosis really made us understand that executive functioning type of complaints were the most prevalent in terms of like what worry is and cognitive function difficulties are. So that's one part of the question. The second part of the answer, I agree with you. It would be, I mean, it's complicated, right? And we know that some of the kids will travel and feel unwell. And so yeah, it's a whole endeavor that could be much easier. And of course, having people all together for a week or so. I mean, we've traveled before to conferences and tested like that, just traveled with all the EEG equipment and set the lab in a couple of hotel rooms. So we've done that. For this project, because it's a multi-day project, we would need a lot of hands to be able to collect a significant number of data. But I'm sure that with a slight different project, that's probably less less data are being collected. We could definitely do that. As you know, I'm not officially involved anymore. So I, I can't answer to that part, but you can talk to Sophie about it.
Hi, thank you for your talk. I was just wondering, with thinking about Dr. Emma's talk right before yours, do you have any data on cysteamine use, age, starting cysteamine, because you have that big age group, 9 to 38.
Yeah.
Do you have data on
So
how that might affect it?
we've collected those data actually. So at the start, I was very quick at showing the overall project because it's just too many things and I didn't want it to be distracting. But we are collecting age of start of treatment. We are collecting GFR. So we are collecting a bunch of things that could help us understand a bit more how those things influence cognitive functioning and brain functioning in cystinosis, but in the future.
Anyone else?
I don't know if this is so much of a question as it is a comment. So I can tell you that it was about 8 years ago that Jake sat in the room and heard one of the talks about functioning and brain and everything else. And he pulled up articles, and those articles have hung above his bed for the past 8 years. I guess my caution to parents is don't expect less. You know, we have twins, one with, one without. Their GPAs are both incredible. They're both capable of so much. So the trap, I think, as a parent, and I think Amy and I struggle with this between the two of us, is just what should you expect? And should your expectations be any less? And if Jake was in this room, he would probably be the first one to grab the microphone and be like, there's nothing that I can't do. And he would not necessarily agree with anything that states anything to the contrary. So I guess to the parents of kids that are younger, I would not change the expectation, I guess is my thought.
Yeah, thank you for that. And I hope that the data that I showed or what I said was not supportive of that in any way, because what we are seeing is not that. You know, we see these slight differences, as I was saying, in terms of brain function, which doesn't mean that they won't be able to accomplish as much as their peers. It just means that some of people living with cystinosis probably struggle with some things that others don't. So they might need a little more time, or they might need some accommodations, but it doesn't mean at all that the end results won't be different from someone without cystinosis. And everyone is different, right? So we see some people struggle a little more with some things and others don't struggle at all. So our data is actually supportive of not many differences in terms of brain response in people with cystinosis.
Okay, thank you, Dr. Francisco.
you.
Appreciate you. Okay, so now we're on break, and then we'll all be down in the salon. Oh, hold on. Nancy's got something to say.